Hello, we are Gabriela, Ethan, Christopher, and Luis, and this is our final nanomaterials and nanobiotechnology project. We will be presenting on nanoscale drug delivery systems. One of the earliest examples of nanomaterials intentionally used by humans was that of the Lycurgus cup. Created in the 4th century and currently on display at the British Museum in London, this cup's glass is embedded with colloidal nanoparticles of silver and gold. This causes the cup to shine a ruby red when illuminated from inside, as opposed to its normal olive green. A few centuries later, in 1955, Professor Horst Jaskowitz, a German biochemist, reported the creation of the first polymer drug conjugate. This polyvinyl conjugate contained a dipeptide spacer between the drug and the polymer, and it is considered as the first nanoparticle for therapeutic use. This technology, along with the discovery of liposomes in the 1960s, marked the birth of nanocarriers. One of the most important findings that aided in the development of nanomedicine delivery came from the work of Japanese pharmacologist and chemist Hiroshi Maeda in his work with colloidal particles of conjugated anti-tumor proteins. The breakthrough finding came from the preparation of nanoparticles coated with hydrophilic polyethylene glycol, or PEG. This allowed for improved nanoparticle stability. It increased the half-life time and blood circulation of the drug, as it reduced the recognition of nanoparticles by macrophages. With this improvement, the drug, instead of diffusing nonspecifically, was able to extravasate into the tumor via leaky vessels thanks to its nanocarriers. Passively targeting nanocarriers first reached clinical trials in the mid-1980s, and the first liposomes and polymer protein conjugates based products were first marketed in the mid-1990s. So, what are nanoscale drug delivery systems? A nanoscale drug delivery system is a technique utilized to protect, transport, and release of biologically active compounds at a specific time, to a specific target site, and in a safe, reproducible manner with the aid of engineered nanoparticles or nanoscale structures. As seen on the image below, a few mechanisms utilized for drug-releasing nanocarriers are diffusion, solvents, chemical reactions, and stimuli-controlled release. Now, all delivery systems enter the body through three main routes: direct injection, which can be local or systemic, inhalation, or oral intake. Once it is inside the body, the loaded nanoparticles begin to interact with the body's circulatory, lymphatic, and immune system. These interactions directly affect the design characteristics needed for the creation of a successful delivery system, which lead us to the main characteristics that define our delivery system. There are four main characteristics that can define a nanoscale drug delivery system. The first one would be size of particle, as the shape and size of nanoparticles affect how it is perceived by the cells within the body, thus dictating their distribution, toxicity, and targeting ability. Surface properties, such as incorporation of targeting ligands, surface curvature, and reactivity, are key elements in designing nanoscale drug delivery systems. Modifying surface characteristics can address problems like overaggregation and can improve the system's stability and reliability, as they affect receptor binding and with it, the actual pharmacological effectiveness of the drug. Drug loading and release in a nanoparticle-based system formulation is dependent on many factors. These include temperature, pH, solubility of the drug, desorption, drug diffusion through the nanoparticle matrix, and matrix erosion. Depending on the need and application, the type of release will differ. Yet, all loading and releasing techniques will focus on allowing the drug to safely travel throughout the body and releasing the drug at a specific time and site. The final step will involve targeted drug delivery. A nanoparticle drug delivery system should be able to reach, recognize, bind, and deliver its load to a specific pathologic tissue. The system should also minimize and ideally avoid inducing damage to any healthy tissue.
Applications. There are many reasons why using nanoparticles for therapeutic and diagnostic agents, also for the advancement of drug delivery. As we know, traditional drugs available now for oral or injectable administration are not always the best. because it requires a carrier system to enhance their efficacy and protect. Also, the efficiency of most drug delivery systems is directly related to particle size to their small size and large surface area. Drug nanoparticles show increased solubility and thus enhanced viability, additional ability to cross the blood-brain barrier, enter the pulmonary system and be absorbed through the endothelial cells of the skin. Cancer therapy. Current chemotherapy is mainly aimed at destroying all rapidly dividing cells, and we can avoid the toxic effects by using nanotechnology with targeted delivery. Micelles and liposomes offer another option for delivery of chemotherapeutic agents. Additionally, micelles are also a great way to make insoluble drugs soluble due to their hydrophobic core and hydrophilic shell. We have dendrimers are highly branched macromolecules with many functional groups available for the attachment of the drug, targeting and imaging agents, and their absorption, distribution, metabolism, and elimination profile is dependent upon several structural features. Also, we have carbon nanotubes, have very hydrophobic hollow interior, water insoluble drugs can be easily loaded them. The large surface area allows for surface functionation and can be done specifically for a particular cancer receptor as well as contrast agents. Another application is for HIV and AIDS treatment. The use of polylactic glycolic acid (PLGA) to prepare nanoparticles in trapping three antiretroviral drugs, ritonavir, lopinavir, and efavirenz. The nanoparticle system yielded sustained drug release for over 4 weeks, about 28 days, while free drugs were eliminated within 48 hours, 2 days. Nanoparticles are known to be able to cross blood-brain barrier by endocytosis, phagocytosis, and many reports exist showing successful delivery of anti-HIV medications. Applications in diagnostic testing. Theranostic nanoparticles can be used for treatment as well as diagnosis. It has been included in many classes of nanoparticles such as drug conjugates, dendrimers, surfactant, aggregates, micelles and vesicles, core shell particles, and carbon nanotubes. If both drugs are combined with an imaging agent in one smart formulation, it is possible to see the pathway and localization of these nanoparticles at the target site, as well as drug action to evaluate therapeutic response. Another application is for nutraceutical delivery. They are commonly consumed as complement to various allopathic treatments, also helps to provide extra health benefits and decrease risk of several chronic illnesses. The majority of nutraceuticals are lipophilic molecules, such as fat-soluble vitamins like A, D, E, and K, polyunsaturated lipids, and other phytochemicals. Nanotechnology once again offers comprehensive assistance, and most of the investigations have been focused on improving the dissolution mechanisms of nutraceuticals via nanoparticle formulations.
The advantages of nanoscale drug delivery systems are numerous. These carrier systems can combine a drug payload with a shell or other vehicle that increases uptake by targeted tissues and cells. An example is a liposomal delivery system, which increases uptake through the cell membrane by absorbing into the endocytic vesicle and launching cargo directly into the endosome. Controlled release of drugs can be accomplished by leveraging material properties and molecular behavior of the payload. Conformational shifts of the molecules in changing environments, enzyme hydrolysis, and stimuli-responsive components are all viable techniques currently undergoing research. Some specific examples will be covered in the next slide. Many bulk solid-based drugs accumulate in the kidneys or liver, which can cause toxicity over time. This slows down elimination and does systemic damage to the body's excretory system and, in some cases, other bodily systems. This occurrence is the result of high doses necessary for therapy due to low specificity and uptake. Nanoscale systems, on the other hand, are often used at lower doses because they are targeted and have improved absorption kinetics. Therefore, accumulation is reduced and the elimination system is uninhibited. Nanoscale drug delivery systems also offer the benefit of design modularity. Techniques developed by chemists can be used to attach specific antibodies, enzymes, ligands, and other materials to the surface, as you can see in the picture on the top right of the slide. This is a process known as targeting or functionalization. The material properties dependent on environmental stimuli can also be used in clever ways to modulate drug delivery. This can be used to tune the system to release its cargo on demand. An example of this is the use of photoresponsive nanogels or nanoscale hydrogels, which can allow UV light pulses to modify the cross-linking of the nanogel and increase or decrease swelling. This in turn modifies the release of the drug into serum or tissues. An additional insight is that these delivery systems can be combined with existing therapeutics to increase efficacy. This phenomenon can be observed in the use of existing chemotherapeutics approved for cancer treatment being used on nanoscale delivery systems. This reduces the complexity of research endeavors because scientists may improve the efficacy of trusted therapies rather than inventing entirely new drugs. Likewise, materials that show promising biocompatibility can be reduced to the nanoscale, as in the case of graphene. These decisions may help reduce the length of time for regulatory approval because it opens up pathways of approval like a 510(k) pathway or a Class 2 approval process by allowing developers of therapies to cite existing predicate therapeutics. Let's review some examples of needs that are currently undergoing investigation. The current standard of care for chemotherapeutics involves the application of cytotoxic drugs in an effort to kill tumor tissues. These cytotoxic molecules contribute to a long list of side effects, with symptoms ranging from a simple migraine to loss of sexual function and sarcopenia. These side effects make this form of therapy dreaded and greatly reduce the quality of life of patients in those programs. Therefore, the use of targeted drug delivery systems can provide a solution to this problem by reducing the amount of circulating cytotoxic molecules and increasing cell and tissue specificity. A specific example of this is the use of nanoscale graphene oxide with a doxorubicin payload. Doxorubicin is a commonly used anti-cancer drug. Conveniently, this molecule can be attached to nanoscale graphene oxide sheets via covalent bonding. These sheets are then functionalized so that they may be targeted at a specific type of cancer cell. When this system is applied to cancer cells, it is endocytosed and carried to the endoplasmic reticulum of cancer cells. When exposed to the acidic environment of a cancer cell's endoplasmic reticulum, which has a pH of about 5, the doxorubicin has a conformational change and is detached from the graphene oxide delivery system. This reduces damage to healthy cells because they are less likely to take up the nanoscale drug delivery systems when they have been functionalized. And if they do, they are not as acidic as the endoplasmic reticulum of cancer cells. Neurodegenerative diseases are another example of a disease that has proven resistant to existing therapeutics, and the drug-related options also interfere with the patient's quality of life. The low efficacy of existing drugs can at least in part be attributed to the challenge of overcoming the blood-brain barrier. Researchers investigating this need are exploring the benefits offered by functionalization of nanoscale drug delivery systems. As you may expect, the use of specific targeting molecules and polymer matrices are assisting in the transport of drugs into neural tissues and are currently undergoing investigation from a highly diverse group of perspectives. More broadly, a need in the domain of drug delivery is to control the release kinetics in a strategic way. Absorption and release can have a complex relationship in the body due to a multitude of variables that influence cell and molecular behavior. The efficacy of drugs can be increased if the external stimuli can cause the release of drugs at the most important time. This application overlaps with smart material investigations, and as you can see in the graphic in the upper right, there are a multitude of approaches using everything from thermally sensitive compounds to flashes of specific wavelengths of light in order to control drug delivery and release.
Now on to some of the current devices used for drug delivery in cancer tumors. I will be covering liposomes, dendrimers, polymeric micelles, carbon nanotubes, and polymer drug conjugates. Liposomes were the first nanoscale drug approved for clinical application. They are spherical, self-closed structures with at least one lipid bilayer and an aqueous center. They are very useful in drug delivery because of their ability to encapsulate hydrophilic agents in the aqueous center and hydrophobic drugs within the lamella. Liposomes modify the lipid layer structure to mimic the biophysical characteristics of living cells. Liposomes are used for in vivo delivery of many anti-tumor drugs such as doxorubicin, paclitaxel, and docetaxel. Doxorubicin slows or stops the growth of cancer cells by blocking an enzyme called topoisomerase, and paclitaxel inhibits tumor endothelial cell growth, and docetaxel inhibits mitosis. Because docetaxel is poorly soluble in water, it is more commonly used in solid tumors, where it improves clinical efficacy without any medication. There are currently 4 approved liposomal products for clinical use, and they have all increased or enhanced delivery to the tumor site and load system toxicity. One major drawback of liposomes is their rapid clearance from the bloodstream. To combat this, hydrophilic polymers such as polyethylene glycol, PEG, is used as a surface coating to extend the time liposomes circulate in the blood from a couple minutes to several hours. Dendrimers are 3D, highly branched spherical structures with 3 components: a central core, interior branches, and an exterior functional surface. Dendrimers have a large number of surface functional groups, so they can quickly and easily bond with targeting agents for selective delivery of drugs to cancer tumors. They can also transport highly hydrophobic drugs because of their cavities used for macromolecule encapsulation. Because of the spherical shape and large amount of functional surface groups, anti-cancer drugs can interact with dendrimers by encapsulation, electrostatic interaction, and covalent conjunction. Polyamidoamine (PAMAM) dendrimers are the most used for tissue engineering, regenerative medicine, and biotechnological applications because of its ease of use, ideal chemical and physical properties, and large number of surface functional groups when compared to other dendrimers. PAMAM dendrimers have a high level of control because they have an inner alkali diadenine core and a peripheral shell made of tertiary amine branches. The only drawback to these dendrimers is their high level of toxicity, which can be easily fixed with surface modifications. PAMAM dendrimers have been studied on several cancer types. One example was done on cervical cancer with a combination of magnetic chemotherapy and hypothermia on HeLa cells. The results showed that cancer cell viability fell from 100% to 3.6% due to the high temperatures and deoxorubicin interactions. But non-cancerous cells were not damaged. The toxicity did not have any effect on them. Polymeric micelles are composed of block copolymers with hydrophilic and hydrophobic parts that self-assemble in an aqueous environment. Polymeric micelles have a low critical micelle concentration, which allows greater structural stability and allows them to retain encapsulated drugs and remain stable after dilution. Their hydrophobic core can be customized to fit the needs of the drug. And lastly, the high molecular weight of the outer shell protects the micelle from phagocytes. Polymeric micelles can be designed to target specific delivery sites by physical, chemical, or electrostatic interactions. Polymeric micelles have proven to be successful in drug delivery, but have shown poor results trying to get into cancer tumors. To solve this problem, polyethylene glycol (PEG) is used in a process called PEGylation, and it improves the solubility of the drug and biocompatibility. This process works because the PEG shields the polymeric micelle, preventing interactions with charged particles or drugs in the tumor, which allows the micelle to get deeper into the tumor. This shielding effect has been used with doxorubicin in a 3D cell culture, and in vivo models to enhance depth in the tumor. Carbon nanotubes are essentially sheets of graphene rings from hybridized carbon atoms that are rolled to hollow cylindrical tubes and are considered one of the strongest materials known. There are 2 types of carbon nanotubes, single-walled and multi-walled. Single-walled nanotubes are single graphene cylinders, while multi-walled carbon nanotubes have multiple complex layers of graphene cylinders. Single-walled carbon nanotubes are small in diameter, flexible, and have a low loading capacity, making them ideal for imaging. On the other hand, multi-walled carbon nanotubes have large diameters, resulting in a higher surface area and higher loading capacity. Carbon nanotubes are gaining popularity in drug delivery because of their unique physiochemical properties. Carbon nanotubes are able to increase the temperature inside tumors by absorbing near-infrared light. They need certain surface modifications, covalent or non-covalent, and they're often needed because the tubes are inherently hydrophobic and have a hard time dispersing in aqueous phases. The cylindrical shape of the carbon nanotube allows it to enter the cell by passive diffusion across the lipid bilayer or endocytosis, where the carbon nanomolecule is engulfed by the cell membrane. Doxorubicin was delivered to cancer tumor cells in carbon nanotubes and showed less side effects and higher drug retention. Paclitaxel and gemcitabine are currently being researched to improve their toxicity and specific site delivery. Polymer drug conjugates are drug delivery systems that incorporate therapeutic agents by chemically linking them to the polymer. The drug delivery system typically consists of 3 units. The first is a water-soluble polymer backbone that ensures biocompatibility and solubility of the conjugate. The second is a targeting moiety that improves delivery of the conjugate to the targeted cancerous cell or tissue. The last component is a therapeutic agent or drug that is covalently bonded to the polymer with a linker, and that linker is responsible for the controlled release of that drug. Polymer drug conjugates tend to have a high molecular weight that extends the time they circulate in the blood, but can also hinder the drug conjugate from reaching the targeted cancerous cell or tissue. Polymer drug conjugates are administered intravenously and reach cancer cells or tissue by passive tumor targeting due to the leaky vascular and poor lymphatic drainage. A lack of a proper lymphatic system allows these nanomolecules to stay in the tumor and release their anti-cancer drugs longer than a fully developed system. To enter the tumor, the size of the nanoparticle must be from 10 to 100 nanometers, and this can be achieved by the EPR effect. HPMA-based polymer drug conjugates linked with anti-cancer drugs such as gemcitabine, doxorubicin, and tyrosinamide have shown to effectively kill cancer cells by preventing new blood vessels from developing and activating apoptosis of the tumor. These polymer drug conjugates did show evidence of microscopic injury to the heart tissue, so they should be used with caution for extended drug delivery. PEG-based polymer drug conjugates linked with platinating agents such as cistaplatin, carboplatin, and oxaliplatin have shown increased anti-cancer activity in advanced solid tumors. They have also been tested on breast cancer, where PEG-based polymer drug conjugates were linked with folic acid and trastuzumab. They showed apoptosis of 80% and tumor regression. Anti-cancer drug doxorubicin was linked with PEG and displayed the longest half-life in the bloodstream, reduced toxicity, high anti-cancer activity, and reduced tumor size. There are some drawbacks with PEG-based polymer drug conjugates. They are not biodegradable, so they are excreted based on the molecular weight, and they also have a poor loading capacity because of the presence of only 2 reactive groups.
Nanometer-sized particles have unique physical, electromagnetic, and chemical properties that are distinct from bulk solids. As you have just heard, this is a rapidly growing field with numerous approaches and opportunities. From diagnostics to therapeutics, nanoparticles have been making a difference since the 1960s. Current nanodrug delivery systems come in a variety of forms: liposomes, dendrimers, polymeric micelles, carbon nanotubes, and polymer drug conjugates. The NDDSs that we have touched on here are being used to investigate new cancer therapies and treatments for chronic diseases such as HIV. The advantages of NDDSs is their capacity for targeted delivery, controlled release, superior absorption, and tunable characteristics that can reduce accumulation and change other behaviors. This technology can change therapeutics by leveraging these options against diseases with a very poor standard of care, such as aggressive cancers or neurodegenerative diseases. When combined with other nanotechnologies, medicine may see a revolution.